So, good evening. My name is Mujmule Vishal Baban. I am now giving a demo lecture for the purpose of faculty position as an assistant professor of life science at IFAS. So, for today's presentation, I have chosen the point of replication of DNA in bacteria, which is this model is actually completely explained in Escherichia coli. So there are two major bacteria in which this semi-conservative model of replication of DNA is given or explained. First in E. coli and the second one is Borrelia. Borrelia is a genus of spirochete. So, what is replication? We will so the flow of this lecture will be like, first I will explain you the small concepts about the replication, what is replication. Then I will give you different models which are explained in replication, then various components like proteins and enzymes that take part in replication process, and then I will give you the complete model explanation of mechanism how the replication of the DNA in prokaryotes takes place. So, what is replication? The word itself means gives the complete meaning. Replication is nothing but the replica of the parent DNA. DNA is nothing but a double-stranded helical structure in which the polynucleotide chains are arranged in helical manner, where the uh, phosphodiester bonds form the backbone of this helical structure, and hydrogen bonds form the uh, what we can say helps in the binding of this helical structure. So the rule is that always adenosine deoxyadenosine triphosphate will get attached to the thymidine deoxythymidine triphosphate, and deoxyguanidine triphosphate will get attached to the guanidine deoxyguanidine triphosphate. So the bond that get formed should be known. In adenine and thymidine triphosphate, the two hydrogens bonds will be get formed always, and in cytidine deoxycytidine triphosphate and deoxyguanidine triphosphate, there will be three hydrogen bonds. This information is important and you should keep it in mind, and I will give the significance why it is important. So I was explaining the double helical structure now. What is double helical structure? Double helical structure is nothing but the polynucleotide chain of deoxy nucleotide triphosphates get formed, and these nucleotides, when are arranged in a helical manner, they are bound together with the help of hydrogen bonds. So what type of hydrogen bonds get formed? So hydrogen bonds formed are given as two hydrogens bond will be formed between adenosine and thymidine, and in three hydrogen bonds will be get formed in uh, cytidine as well as guanidine. So in next structure, so this is nothing but the parental DNA. This is parental DNA, and in the following diagram you can see that this parental DNA gets replicated in two daughter DNA, and this daughter DNA is nothing but of semi-conservative nature. This concept is highly important because there are various questions that may get formed that this why we say that this model is semi-conservative in nature. Then second concept is that this is also called bidirectional. Why it is called bidirectional? The this concept and these words you should get completely by hearted, and you should clearly know these concepts. So why semi-conservative? So there is a simple model or experiment. It is called Michelson and Stahl experiment. The semi-conservative nature, the bidirectional nature, and Michelson and Stahl experiment are some of the important points that get asked in DBTs, DBT JRF, CSIR NEET, GATE exam. And so now I will explain why it is called semi-conservative in nature, and what is Michelson and Stahl experiment? So Michelson and Stahl experiment is based on centrifugation. So there are 2 points that the nitrogen, heavier nitrogen and another isotope which is light nitrogen are used. N14 and N15 are used. So in this experiment, the parenteral bacteria are grown first with the help of N15 isotope, which is a isotope which has high weight, because of which when parenteral bacteria will be get grown in the nitrogen 15, this will have a DNA which will have nitrogen 15, which will have higher molecular weight, and because of which in the next part of the experiment we will going to be use the centrifugation process with the help of copper chloride as a medium. And in this experiment it will be you will understand that this nitrogen 15 is an important factor that will help us understand why this experiments gives the semi-conservative nature of the DNA replication in prokaryotes like Escherichia coli as well as Borrelia. So the generation 0 or G0 will be grown in a medium which will have the nitrogen 15 as a nutrition medium. The nutrition medium will be such that the nitrogen 15 will be present as a source of nitrogen, and after that we will extract the parental bacteria or generation 0, and then we will take them into the medium which will have nitrogen 14. So this is Michelson and Stahl experiment, and in which then we will grow the generation first, and when generation first will be grown in the nitrogen 14 containing medium, we found that there was the proportion of DNA which we get to found is that the half of the DNA or the bacteria containing the DNA is of hybrid type. and other hub of the DNA will be of higher density. Because half of the DNA will be hybrid and half of the DNA will be of higher density, then this G1 or generation first, when again we will go for a growth in nitrogen 14 as a G2 or generation 2, it was found that the the amount of hybrid containing hybrid, or we can say nitrogen 14 as well as nitrogen 15 containing nitrogen 14 and nitrogen 15 containing DNA was increased proportion of light DNA. Light DNA will be of having higher amount of nitrogen 15, will be containing nitrogen, higher amount of nitrogen 14. In this there will be no amount of nitrogen 15 or less amount of nitrogen 15 containing DNA. So I will again mention that बंद नको केलेलं आहे का? So what will happen in G1, G0 and G2? G1 there will be high density DNA. It will be analyzed through the centrifugation process. Half of the DNA in G1 was of high density and half of the DNA was of hybrid density, or in between nitrogen, because it was combination of nitrogen 14 and nitrogen 15 containing DNA. And in G2 the higher amount of light DNA will be formed, higher proportion of light DNA. Least amount of we can say the high density DNA and a significant amount of hybrid amount of DNA. and each generation like G1, G0, G1, G1, G2 was of time which will help the culture double its amount. So what was the conclusion, or what was found through this experiment? It was found that the nature of the replication model is semi-conservative. Though parental DNA gets when the parental DNA gets replicated, when the parental DNA gets replicated, the half of the amount of DNA will get divided into each daughter culture. So here is the replication fork. This is nothing but the position at where the actual replication process is going on, and now I will let you know the exact way or the model by which the replication takes place. So we all know that the DNA or the nucleic acid present in prokaryotes is of circular nature, and in this circular or sphere or type of nucleic acid, there is a single point of origin or origin of replication. This point is to be of high significance, because there is always some questions from in CSIR NEET as well as in GATE exam. So origin of replication, in books you will find this point to be denoted by ORIC, ORIC, or origin of replication. So what is ORIC? ORIC is nothing but origin of chromosomal replication. C stands for chromosomal. This is origin of replication. Origin of replication is nothing but the point where there is high attraction for DNAA. DNAA is nothing but a type of protein. Never get distracted or understand it to be nucleic acid type. It is a protein which helps to start or commence the replication. I will note down all the proteins as well as enzymes which take part into this process. So DNAA gets attracted to this particular point of replication or origin of replication or origin of chromosomal replication, because it has some specific type of nucleotide sequence. And the second type of characteristic which this origin of replication holds is nothing but it has high amount of A and T or AT nucleotides. And you should now remember that I have clearly mentioned that A and T has 2 hydrogen bonds. What is significance of this information? Why had made you remember this? Because this AT in comparison with CG or deoxycytidine triphosphate and deoxyguanidine triphosphate, it will form 3 hydrogen bonds. And breaking of these hydrogen bonds by the enzyme helicase. Helicase is nothing but the enzyme which will start the replication process. DNA helicase. It will be highly simple for helicase to ligate or lies these two hydrogen bond containing or adenine and thymidine containing space or polypeptide chain or this particular type of pattern in this origin of replication, then C and G containing 3 hydrogen bonds, as this will require higher amount of energy, and DNA helicase takes its energy by use of ATP, adenosine triphosphate. So why bidirectional? I made you remember that there is nothing but a origin of replication. This is origin of replication. When origin of replication will get formed, there will be higher amount of DNA that will get attached. There will be DNA that will get attached to this position. DNA is nothing but helicase enzyme. There will be higher amount of DNA also a protein which helps DNA to get attached at this origin of replication. So when origin of replication will get formed, this will form replisome. The structure is called replisome. The structure containing all these enzymes and proteins which will get formed with the holoenzyme DNA polymerase 3, which is called as replisome. So why bidirectional? When this DNA, this is point of origin of replication, this will form the replication forks, and these replication forks will go in opposite directions from the point of origin of replication. So two replication fork will get formed. This is also replication fork. Replication fork will be get formed when all the type of proteins as well as enzyme which should get attached to this and form a complete enzyme structure which will help the replication process to go on. And I told you that why replication fork? So these will go on in opposite directions. This is point of termination or termination. We can simply say it as termination. Because this process just go on in opposite direction from the point of origin of replication, this is called as simply the bidirectional nature of the replication of DNA in prokaryotes, or especially with the example in E. coli. So now I will list you the all the enzymes and proteins which take part in the replication, and then I will give you the mechanistic procedure how this replication process goes on. Proteins and enzymes in DNA replication. Remember the semi-constitutive nature and bidirectional nature of this replication process. This is in prokaryotes. Don't forget. So I will first list DNA. Remember, I am writing this as D and small in small letters NAA. So this protein is nothing but protein and initiates this initiates the process of this will initiate the process of DNA replication because at the origin of replication this will be the first protein that will go and bind. This will be rich in AT polynucleotide and chain. The polynucleotide chain here will be with higher amount of significant amount of AT, as well as there will be also some polynucleotide patterns, unique sequences which are highly regarded in starting or for the starting process of this replication, but is the point which will be taught later on. So DNA will go and bind. So next protein is DNAB protein. Proteins are also enzymes. You should know that RNAs are also enzymes, but they are called ribozymes. So DNAB, this has helicase nature. This will be the first enzyme which will help nick the double stranded nature. The nick will be done here. There will be given a cut with the help of helicase nature. There will be lytic activity or bond breaking activity with the help of energy which is obtained from ATP, also shown by ATP slash nucleotide adenine dinucleotide containing a positive charge. It uses the energy by breaking the pyrophosphate bond. This energy will be two phosphates will be get formed by a pyrophosphate, and there will be also energy released, and this energy will be used by our lytic nature having protein or enzyme which is DNAB. So third protein is DNAC protein or enzyme. What is the significance of this protein? Why it is? What role it plays? So it helps in binding of DNAB having a helicase nature at the origin of replication. Helper protein for DNAB. Remember the nature of this proteins enzymes. So there will be DNA polymerases. So there are 5 types of DNA polymerases. I will let you know for sake of time 2 which are major and which I will let you know at this point are there are 5, 1 to 5. These also have a significance in this process. DNA polymerase 3 and DNA polymerase 1. So DNA polymerase 3. What is DNA polymerase 3? This is the like hero of this process. This enzyme is one which will do all the work. All the mechanism done is by DNA polymerase only when the process will start. Before that there will be many proteins which will help it to initiate the process. So what is DNA polymerase 3? It is an holoenzyme. Holoenzyme are formed by coenzymes and apoenzyme. So DNA polymerase 3 is a apoenzyme which will require some proteins and enzymes which will coordinately help it in doing the replication process. And there will DNA polymerase 1. What is DNA polymerase 1? This is nothing but ribonuclease. Remember what is nuclease? Nuclease is nothing but having a lytic process. So then DNA ligase, DNA ligase is a protein or enzyme which will have a ligating or binding action. This will help the double-stranded DNA helical structure to ligate at some points in the process, and I will mention it as well. So DNA topoisomerases. So DNA topoisomerases. DNA topoisomerases are like enzymes having double activity. These have activity of lysis as well as ligating activity. It will cut as well as bind. And the most important example in DNA topoisomerases is DNA gyrase in E. coli. GYRASE, DNA gyrase. Then this process will also contain some enzymes like ribonuclease H, ribonuclease H will be having the activity of same as of DNA polymerase 1. Same activity. This will help nicking out the or cutting out the RNA primers from the Isozaki fragments as well as the primer which is used in the DNA replication process at the first point. So ribonuclease, and there will be a enzyme. So these are all the enzymes which will be used through this process. So then what is the mechanism? So I will explain you the mechanism now. So DNA polymerase 3. DNA polymerase 3 is made of tau type of complex. Tau type of clump complex. So tau complex will be having a binding nature. So I will make the diagram for this. So this is holoenzyme DNA polymerase 3. So this is tau complex. These are 3 appendages. These will be 3 appendages, and then what are these? These are formed from 3 polypeptide chains, and these can be given as alpha, epsilon, and theta. And this is beta polypeptide. So after that, how the process of DNA replication will take place? This will be DNA polymerase 3. What is DNA polymerase 3 is made of? Tau complex, beta complex, alpha, epsilon and delta complex. So remember this nature. So two of these appendages and this structure will be engaged in replication for one of the polynucleotide chain of the double helical structure of the DNA, and one will be for this two will be engaged in for lagging strand. I will explain what is lagging strand. And one will be engaged for leading strand. For leading strand. So how this process will go on, let's know it. When origin of replication will get formed, DNA A, DNA B, DNA C will be get bound to each replication fork, and when this will get bound to the each replication fork, the replication process will go on, and it will be bidirectional in opposite directions in two replication forks, and it will be in bidirectional and semi-conservative type. This will form a structure. You should be remembering it will form theta structure. Remember this word theta. So I will again draw the DNA polymerase 3. These are alpha, epsilon and delta polypeptide chains, beta clamp, and this is tau clamp complex, tau binding complex. So the parental DNA will enter from which position? It will enter from this position. One of the strand will go through this way, and here it will get formed again into daughter DNA of double-stranded and helical structure. This is leading strand. I will mention why leading strand, why lagging strand. These are also some important concepts. And then one of the strands will go through this pathway, through two complexes of epsilon, alpha, epsilon and delta. This is beta complex, and the polypeptide chain here formed will be of lagging nature. Of lagging nature. Of lagging nature. Lagging strand. So DNA polymerase 3 complex. Our parental DNA got entered from this position in this direction. It moved to this way This is leading strand, this is lagging strand, and the process will go from 5 prime to 3 prime. Remember, these are nothing but the position of carbon. 5 prime to 3 prime, and replication will also go in same direction, except here the replication will take place 3 prime to 5 prime on the lagging strand. So when this process will get start, the process will get start when at the origin of replication the lytic nature containing DNA helicase having the nature the protein enzyme DNA B will get bind, it will lyse the double helical structure, and when it will lyse the double helical structure, the double helix will get opened, it will get the single stranded DNA polypeptides will get bound by single stranded binding proteins, and these are called as SSB proteins. This helps in stabilizing these proteins. Then in next point, what will happen? The each strand will get this strand will get entered. So there will be, here will be DNA helicase activity. Here will be DNA primase. Why DNA primase? DNA primase because the replication process of DNA can't start directly by adding a deoxynucleotide, because there is no deoxynucleotide accepting nucleotide or position like 3-hydroxy. There should be presence of a 3-hydroxy group that will first accept the 5-phosphate group of incoming deoxynucleotide, which is not present in this at the start of the process. So there will be DNA primase which will add a RNA primer. So at the first there will be addition of RNA, a piece of RNA which will be RNA, and after this RNA last nucleotide or at the growing phase of this RNA, there will be 3-hydroxy. This 3-hydroxy will accept by the polymerase nature of this DNA polymerase 3, it will accept the a phosphate group of upcoming deoxynucleotide, and so by this process the DNA polymerase activity will go on. So leading strand and lagging strand, how this go on? DNA polymerase 3 complex. DNA polymerase 3 complex. This is incoming DNA. This is replication 4 going in this direction. This is parental DNA. This is leading strand. Let's give it a helical structure. Here will be RNA primer which was added. No, not here. At this position by DNA primase, and then this leading strand will go on growing. But there are in the lagging strand in 3 to 5 direction, 3 prime to 5 prime, the growth will be take place such that there will be formation of lagging strands of DNA on new RNA primes, RNA groups in 3 to 5 direction, and these are nothing but Okazaki fragments. Okazaki fragments. At the later stage, these Okazaki fragments will get bound with the help of ribonuclease H. This ribonuclease H will cause ribonuclease H. This ribonuclease H will help this DNA to get into its complete nature by nicking off or lighting off the or cutting off the RNA primers, and addition of deoxynucleotide triphosphate at these spaces which will get formed, and this whole work will be done with the help of DNA polymerase 1. DNA polymerase 1. DNA polymerase 1. So these are called Okazaki fragments after the great scientist Rizi Okazaki, and these are called this is nothing but the leading strand. It will be completely, totally a continuous strand, and because of this it is also called as continuous replication here, but this continuous replication at the lagging strand, discontinuous replication. So at the end there will be formed when these two forks containing at the first point there will be the primosome formation, lighting activity, two forks will be formed that will move in a bidirectional format to replication for forks moving in bidirectional format directions and to the terminal point. This is called termination. When this will reach termination, a protein taus will bind and help this DNA polymerase 3 to go away, and two daughter nucleic acids DNA or circular DNA will be formed, daughter DNA. This is nothing but daughter DNA. So there are at the last the two problems which will get formed in this during the replication. When these replication force will be moving forward with the help of DNA polymerase 3, it will have helicase nature, and because of which supercoiling will get formed at this position in front of this, and this will be removed with the help of topoisomerase. With the help of topoisomerase. Remember this word, topoisomerase, and this is also called as gyrase. So topoisomerase or DNA gyrase. In topoisomerase, this will help this molecule to go away. So this will have the lysis activity and then ligate again and remove the removal of supercoiling. This will remove the supercoiling that will get formed at this position at the in front of the DNA polymerase 3, which will be moving with the helical nature. So after this, there will be at the last 2 points there which will be get formed. There are two problems, that is catenins will be get formed, and there will be formation of dimerized RNA, DNA, sorry. So the catenins will be removed with the help of nuclease activity. This will nick off here, open the circular DNA, and it will remove this, and again having the ligating activity bind it, and individual daughter nucleic acids will be get formed. And here the remember ribonuclease